and Mathematics International Conference 2022. Welcome to parallel session in room 16. We have 13 speakers, including one invited speaker, but unfortunately, Professor Dr. Jalifa hasn't joined yet. So we started the presentation from the first speaker, ABS 120 by Ibu Yusi Pratiwi.
Everyone, introduce my name Ayewinani Pangastuti as a speaker in an article entitled Analysis of Phosphor Using Ammonium Molybdate with thiourea and hydrogen sulfate as reductors by UV spectrophotometry methods. Phosphorus is found naturally in peanuts because it is sourced from the soil and fertilization to increase the growth and development of bean plants requires phosphate fertilizer. Excessive use of phosphate fertilizer can increase the concentration of phosphate in pea plants and increase the phosphate content in the soil. Increased concentration of phosphate in peanuts is harmful to human health if consumed by humans if it exceeds the threshold of 750 until 1250 milligrams per day. Excess phosphate can cause to high phosphate in the blood of hyperphosphatemia based on the dangers that can be caused by excess phosphorus in the body. A phosphorus analysis method using the UV spectrophotometry method was carried to ensure food safety. In the research that was carried by Soji in 2003 on the determination of phosphorus by the formation of a complex between vanadium molybdophosphate and malachite grain, the drawback of this method involved the extraction process. Therefore, research on phosphorus analysis has been carried with the formation of a blue phosphomolybdate complex, where the principle is based on the oxidation reduction reaction between ammonium molybdate and thiourea and hydrogen sulfate as a reduction agent which can be measured with a UV spectrophotometer and validated by analytical methods with linearity, sensitivity, LOD, LOK, precision, and accuracy parameters. UV spectrophotometry analysis methods of phosphorus was carried by reacting a solution of potassium dihydrogen phosphate with a solution of ammonium molybdate in an acidic environment and added with a reducing solution such as thiourea and hydrogen sulfate. The mixture of the solution formed a blue-colored phosphomolybdate complex whose absorption was measured by UV spectrophotometry. Further optimization of phosphorus analysis conditions such as determination of maximum wavelength, optimization of ammonium molybdate concentration, and optimization of thiourea and hydrogen sulfates reducing concentrations. The optimization results obtained were validated by the analytical method with parameters of linearity, sensitivity, LOD, LOK, precision, and accuracy. After validation of the method, the phosphorus content in the peanut sample was determined. Result: optimization of the analysis condition on the determination of the maximum wavelength measured by the absorption of the complex compounds phosphomolybdate in the visible area in the range of 5000 until 800 nanometers. The result was a wavelength of 689 nanometers. Next, optimization of the concentration of ammonium molybdate solution obtained is 0.0115 molarity. If the concentration exceeds 0.0115 molarity, the absorption will decrease because a new complex will occur, namely PMO. Next, Optimization of the concentration of TiO-urea solution obtained is 0.6568 molarity. If the concentration exceeds 0.6568 molarity, the absorption will decrease because a further reaction will occur. Optimization of analysis condition in hydrazine sulfate. Determination of the maximum wavelength of time is 689 nanometers. Next, optimization of the concentration of ammonium molybdate solution obtained is 0.01 molarity. Next, optimization of the concentration of TiO-urea solution obtained is 0.1 molarity. Validation of analysis method. For hydrazine sulfate reducer gate, linearity is the correlation coefficient of 0.9984, which is linearity sets that the relationship between concentration and absorption. Sensitivity is the response of the instrument to the method used and expressed by the molar absorptivity obtained of 7.92 multiplied by 10 to the 4 liters per mole centimeters. LOD is the smallest limit test parameter owned by the tools or instruments. LOK is the lowest concentration of analyte that can be determined. Until the LOD and LOK values are equal to 5.26 multiplied by 10 to the minus 4 milligrams per liter and 1.59 multiplied by 10 to the minus 3 milligrams per liter. Precision is the result of repeatability by the same tools and analysis obtained for 1.25%. And accuracy is expressed by the percent recovery obtained between 90% until 117%. For the validation of the TiO-urea analysis method, the linearity of 0.9961 look at from the validation of the analysis method carried out that the beta reducer is hydrazine sulfate. Furthermore, the determination of phosphorus level in peanut is carried out. Conclusions. Optimum conditions for phosphomolybdate complex in the TiO-urea reductor and hydrazine sulfate obtain the maximum wavelength of 689 nanometers. Phosphorus analysis method using TiO-urea and hydrazine sulfate reductor by UV spectrophotometer has qualified the validation requirements analysis methods. Hydrazine sulfate reductor has better linearity, LOD, LOK, and precision in reducing complex phosphomolybdate compound. The result of analysis of the phosphate content in peanut does not exceed the food safety quality standard according to the Indonesian Nutritionist Association in 2009. Thank you.
Okay. Thank you for the nice presentation. Is there any questions from the audience to our first speaker?
May I ask the questions, Bu Vera?
Okay, please, Ayu. Oh yeah. Please, you can unmute your speaker, your microphone. Yes.
Iya,
Please,
baik Bu.
Bu Irma. Yeah.
Okay. Oh, can I
Yeah.
start?
Yeah.
Yes.
Please, Bu Irma.
Okay, thank you for the nice presentation. May I ask you second question, two questions, please? So first of all, I want to know why some nuts contain phosphorus compound, right? And then the second one, you use 3 methods, molybdate and Yeah, you use three methods to determine the phosphorus contamination. So why you use, I mean, the reason why you use three methods, three different methods, and the explanation, please. Thank you.
Thank you for the question, Miss Irma. Yes, phosphorus is found naturally in peanuts because it is sourced from the soil, and fertilization to increase the growth and development of bean plants requires phosphate fertilizer. Excessive use of phosphate fertilizer can increase the concentration of phosphate in pea plants and increase the phosphate content in the soil. Increased concentration of phosphate in peanuts, it's harmful to human health if consumed by human if it exceeds the threshold of 750 until 1,250 milligrams per day. Excess phosphate can cause to high phosphate in the blood of hyperphosphatemia. Based on the dangers that can be caused by excess phosphorus in the body, a phosphorus analysis method using the UV spectrophotometry method was carried out to ensure for safety. In this method, the used because thiourea is thiourea and hydrogen sulfate is stable and easy to use. Thank you.
So from the conclusion, how is the concentration of peanut? You use three kind of nuts, right? Cashew nuts, and then almond, and then one is yeah. How is the result of the phosphorus concentration in these peanuts?
In this method, free samples can be applied, so there are no special sample.
So is it safe to be consumed?
Yes.
Okay, thank you. Thank you for the explanation.
Thank you, Ma'am Irma.
Thank you, Irma, for your questions, and thank you, Ayu, for nice explanation. And then we continue to our next speaker, Sylvia Afriani. Okay.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, let me introduce myself. My name is Sylvia Afriani from Chemistry of Universitas Negeri Jakarta. On this occasion, I will present a research entitled Photopolymerization of Product Blending Methyl Methacrylate with Reactive Mesogen RM257 using the UV cooling method. The discussion this time include background, method, result, and discussion. Development of liquid crystal research is still very limited in Indonesia. This is the basic for adding and developing the use of liquid crystal in various industrial fields. Liquid crystal are widely used in all kinds of display devices, such as computer monitor, laptop screen, television, sensor, and navigation system. Crystal are material that have properties resembling crystal or solid, but the physical in the form of liquid. A liquid crystal monomer with a polymerizable end group is called a reactive mesogen. Reactive mesogen can be combined with polymer to form liquid crystal network, and usually these combined polymer are thermoplastic, such as polyamide and polymethyl methacrylate. The combination of liquid crystal and PMMA will produce a liquid crystal polymer that can be formed in the form of the thin film with a thickness of 10 to 100 nanometer. Thin film has advanced such a flexible and lightweight properties, so it can be easily applied in the industrial sector. One method that can be used to form liquid crystal in the form of thin film is UV curing. UV curing is a process that uses high intensity ultraviolet light to initiate photochemical reaction that can produce polymer network in liquid crystal. In this study, the manufactured thin film of methyl methacrylate combined with reactive mesogen RM257 was carried out using the UV curing method with variation in curing time. The liquid crystal polymer MMA RM257 will be characterized using several instruments, namely FTIR, SEM, XRD, and LCR method. The tools used are Becker glass magnetic stirrer, hot plate stirrer, UV curing machine, thermometer, gas plate, spatula, analytic balance, aluminum foil, and drop pipette. Which materials such as MMA, reactive mesogen RM257, DMF, chloroform, and BPO. And the instrument used are FTIR, SEM, XRD, and LCR method. Preparation of PMMA and RM257. First, 3 grams of methyl methacrylate was dissolved in a co-solvent consisting of DMF and chloroform in a ratio of 7:3. The dissolvent was carried out in a 30 ml glass beaker at room temperature, and a 30% awake polymer solution was produced. The same thing was done on RM257 powder. MMA solution was mixed with RM257 solution. BPO initiator as much as the tip of the spatula was added to the MMR and RM257 mix at the temperature of 95 degrees Celsius for 5 minutes and speed of 250 RPM. After that, it will be placed on a glass plate and polymerized using the UV curing method at a temperature of 120 degrees Celsius with varying time, 5, 10, 15, 20 minutes. After that, characterization was carried out using FTIR, SEM, XRD, and LCR method. Result and discussion. MMA and RM257 solution will react and polymerize with each other. Polymerization began when the initiation, where MMR and RM257 will form free radical through the addition of BPO. BPO will break the double bond addition in MMR and RM257. MMI will turn into a polymer, namely PMMA. After that, it enters the second stage, named the propagation, where PMMA and free radical RM257 will react to form cross-linked polymer chains. This process occurs continuously, closed by the termination. At this stage, the polymer can grow stop until the molecule is stable. This picture is the result of polymerization of MMA and RM257 with variation in UV curing time. It can be seen that the sample with UV curing time of 5 minutes has a more transparent color than other samples. But the texture is very susceptible to tearing and is difficult to remove from the substance. The texture gets strong along with the length of the UV curing process. The sample with a UV curing time of 20 minutes has a strong texture, breaks easily, and is less elastic. This is because the curing time is too long, causing the sample to atrophy. FTIR. FTIR is used to determine the chemical function group in the sample. This vector is the result of the FTIR test on the MMA RM257 thin film. The absorption peak ever at 2,950, indicating of CH3 bond. 1,721, indicating of CO double bond. 1,604, indicating of aromatic functional group. 1,248, indicating of COC bond. 1,145 and 1,060 indicate of CO bond. In this spectrum, there is no absorption peak at 1,670, which indicate the absence of a CC double bond. Besides that, the instructance by the absence of an absorption peak at 3,100, indicating the absence of an phenyl belonging to MMA. So it can be concluded that MMA has polymerized to form PMMA and binds to RM257. Based on the graph of the absorption peak in this picture, it can be seen that the spectra of the four samples have a set that's similar or not much different. This indicates that the time difference in this UV curing process does not affect Weyerhaeuser number, but slightly affect the intensity value of the FTIR spectrum on PMMA RM257. Next, SAMS. SAM is used to see the surface topography of a sample. The magnification used are 1,000, 5,000, 10,000, and 15,000. It can be seen that the variation of the curing time of the mixed MMA RM257 thin film cause morphological difference in the four sample shape of the bar scenes in the first sample is the typical molecule shape of reactive mesogen. Sample with a curing time of 50 minutes had the best uniformity and smoothness compared to other sample. This indicates that RM257 has filled the void containing in the PMMA matrix perfectly, while in sample with a curing time of 5 minutes and 10 minutes, RM257 has not occurred the PMMA matrix, well so that the surface is not too smooth and cause the reactive mesogen molecule to be spaced. In sample with a curing time of 20 minutes, the fineness level decreased and caused agglomeration. This was because the sample was specified because the curing time was too long, so that the thin film became like a glass. Next, XRD. Sample characterized by XRD are thin film with UV curing time of 15 minutes. The sample was chosen because it has the best SIM result. Measurements are made at angle range of 10 to 90 degrees. Based on the picture above, there's a shift in the diffraction pattern and increase in intensity. It can be seen that there are wide peaks in the angle range of 10 to 38, indicating the amorphous phase. Sharp peak with the moderate intensity appear at 30.19 and 16.83. Then sharp peak with high intensity appear at 44.66, which indicate the crystalline phase. Before the result is the resulting thin film is semi-crystalline. Characterization of electrical conductivity is known by measuring the resulting thin film using an LCR method. The measurement will produce the conductance and capacitance value of the thin film. Based on the data in this table, it can be seen that the thin film with a curing time of 20 minutes had the highest conductivity value compared to other sample, but the variation in curing time did not significantly affect the conductivity value of the resulting thin film. With the conductivity value obtained, the thin film is classified as a semiconductor material. And conclusion. First, thin film MMA with RM257 was successfully polymerized by UV curing method. Second, the variation time of photopolymerization with UV curing didn't affect the FTIR result. Third, the result of SAM characterization show a sample with photopolymerization time of 15 minutes had a high level of smoothness, and a sample with photopolymerization time of 20 minutes had agglomeration. XRD characterization show that PMMA and RM257 has a wide peak at angle range 10 to 38 subpeak with SMADI intensity in 13.19 and 16.83. Subpeak with intensity high appear at 44.66. Signify that the thin film are semi-crystalline. 5. LCR measurement results show that the thin film with a curing time variation of 20 minutes has conductivity value the highest. 6. Variation in curing time doesn't affect conductivity value significantly. 7. The resulting thin film is semiconductor. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you so much for your nice presentation. Yeah. There will be one question from Eva Handayani.
Yes, okay, thank you, Sylvia, for your presentation. I want to ask one question. Why can RM257 and PMMA react? And how did the reaction occur? Thank you.
Okay, Eva, thank you for the questions. RM257 is a liquid crystal whose end group can be polymerized, which is a so-called reactive mesogen. While PMMA is a thermoplastic polymer. Based on previous research, the combination of reactive mesogen and polymer will form thin film. While RM257 can improve the mechanical property of PMMA, RM257 will experience bond breaking double bond at the end group, and NNI will experience CC double bond and breaking at C central. Bond of which will form free radical, and bond to form can cross-link polymer bond.
Okay. Thank you for your explanation. Okay. Let's move to our next speaker. The speaker is Ibu Yusi Pratiwi, already here.
Good morning everyone. Introduce myself. My name is Aulia Natasha. As a presenter on article titled Determination of Phosphorus Torque Formation of Phosphomolybdate Complex with Tin 2 Chloride and thiourea as reductor by UV spectrophotometry. Phosphor occurs naturally in vegetable because phosphor can be sourced from phosphate fertilization. If the fertilization of phosphate fertilizer is not controlled, it will cause the accumulation of phosphate in vegetable. The entry of phosphate into the human body through the food chain. If the phosphate in the body is too much and exceeds the result of 750 until 1,250 milligram per day, it will cause hyperphosphatemia or kidney failure. Therefore, a phosphorus analysis is needed to ensure food safety. Based on the research conducted by Ritonga in 2012, the determination of phosphate using UV spectrophotometry using ammonium vanadate reagent has drawback, namely the presence of other elemental disturbance that cause the validation result of the phosphorus analysis. Method to form a blue heterophospho-molybdate complex where the principle is based on the oxidation reaction reduction of phosphorus with ammonium molybdate in an acidic atmosphere and then reduced with tin to chloride and thiourea reductor agents whose absorption was measured by UV spectrophotometry and validated by analysis method and linearity parameter, sensitivity, limit of detection, limit of quantification, precision, and accuracy. Research purpose 1. Obtaining optimum condition for phosphorus analysis using tin to chloride and thiourea reductor. 2. Validation the phosphorus analysis method with tin to chloride and thiourea reductor based on linearity parameter, sensitivity, limit of detection, limit of quantification, precision, and accuracy. 3. Obtain information about reductors that are more effective for reductor phosphomolybdate complex. And then determination the rate on vegetable sample. Next method, phosphor analysis method by UV spectrophotometry was carried out by reacting a solution of potassium dihydrogen phosphate solution of ammonium molybdate in an acid atmosphere and mixed with a reductor solution, thiourea, and tin to chloride. Format a blue heterophospho-molybdate complex solution, then the absorption was measured using UV spectrophotometry. The result of the optimization carried out the maximum wealth, concentrasi reductor thiourea and tin to chloride, and concentration ammonium molybdate. The optimization result obtained were validated by analysis method with parameter linearity, sensitivity, limit of detection, and limit of quantification, precision, and accuracy. And next, result and discussion. The first, optimization condition analysis. Optimization of the analytical condition on tin to chloride, determination of the maximum wavelength measured by the absorption of the complex compound heterophospho-molybdate in the visible area in the range of 500 until 800. The result was a wavelength of 729 nanometers. And next, optimization of the concentration of ammonium molybdate solution is 0 obtained 0.032 molars. If the concentration exceeds 0.032 molars, the absorption will decrease because a new complex will occur, namely P2MO18 or 62. Next, optimization of the concentration of the tin to chloride solution, optimum at a concentration of 0.005 to 5 molars. If the concentration exceeds 0.00 to 4 molars, the absorption will decrease because a future reaction will occur, namely a decrease in intensity because Sn plus 2 turn into Sn plus 4. And next, optimization of analysis condition in thiourea. Determination of the maximum wavelength obtained is 689, and optimization of the concentration of ammonium molybdate solution at a concentration of 0.01, and optimization of the concentration of the thiourea solution optimum at a concentration of 0.6 molar. And next, result and discussion. Validation of analysis method. Validation of analysis method for tin-to-chloride reductor get linearity for tin-to-chloride get to 0.9983, and linearity is the correlation coefficient of which is linearity status that the relationship between concentration and absorption. And next, sensitivity is the response of the instrument to the method used and expressed by the molar absorptivity obtained of 0.93 liter per mole per centimeter. And next, limit of detection is the smallest limit text parameter owned by the tool of or instrument. And limit of quantification is the lowest concentration of analyte that can be determined until the LOD and LOQ value are equal to 1.41 milligram per liter and 4.28 milligram per liter. And next, precision is the result of repeatability by the same tools and analysis obtained for one of 10.51%, and the accuracy is expressed by the percent recovery obtained between 1990 of 36% until 109 of 10.08%. For the validation of the thiourea analysis method, the linearity of 0.9961, sensitivity is 2.93 liter mole per centimeter, limit of detection and limit of quantification is 5.62 milligram per liter, 1.7 milligram per liter, and precision 1.30%, and accuracy between 90 3.02% until 110.52%. Look at from the validation of the analysis method carried out that the beta reductor is thiourea. Furthermore, the determination of phosphorus levels in vegetables carried out is phosphorus level in vegetables. Phosphate levels obtained in corn vegetable were 6.034 mg per gram, and phosphate level for broccoli sample were 8.078 mg per gram, and in beans samples obtained phosphate level were 7.086 mg per grams. And next conclusion. 1. Optimum condition for heterophospholipid complex in the tin 2 chloride reductor obtain the maximum wavelength of 709 729 nanometer, and the wavelength of the thiourea reductor at 689 nanometer. And 2. Phosphorus analysis method using tin 2 chloride and thiourea reductor by UV spectrophotometry has qualified the validation requirements analysis method. 3. Thiourea reductor has better sensitivity, limit of detection, limit of quantification, precision and accuracy in reductor complex phosphomolybdate compound. And 4. The result of the analysis of phosphorus content in corn vegetable was 6 obtained 0.34 mg per grams. Broccoli vegetable was 8.078 mg per grams, and in beans vegetable samples of 7 obtained 0.86 mg per grams. So that the phosphorus content of vegetable doesn't exceed the food safety quality standard according to the Association of Indonesian Nutrients in 2009. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. So there will be one question from Miss Sylvia to
Yes. Hello, Aulia. I wanna ask, why in analysis phosphorus has to be reduced with urea and SnCl2?
Okay, thank you. Question, Sylvia. Because if is still in the yellow complex and is not reduced, there are still other disturbance from the organic elements, so that the reduction is carried out, so that there is not interference with other elements. Thank you.
Is it clear enough, Sylvia? Okay, let's move on to the next question from Bu Irma.
Okay, thank you, Bu Vera. Thank you for the nice presentation, Aulia. Could you explain to me, please, why the accuracy is more than 100%?
Sorry, Bu Irma.
Please explain the reason why the accuracy more than 100%.
Because ada akurasi.
You can speak
Because
in bahasa.
akurasi has interval, Bu Irma. Range-nya, range akurasi 80% until 110%.
Biasanya maksimalnya, the maximum value of the accuracy usually 100%.
Iya, Bu Irma.
It means that deviation standard is too big.
Deviation standard.
No. If you're more than 100% accuracy, it means your deviation standard is too big.
But persen akurasi maybe batasnya sampai 110, Bu. Makanya standar deviasinya begitu.
Ya, standar deviasinya jadi terlalu besar. Ya udah. It's okay, Bu Vera.
Okay, thank you, Bu Irma, and thank you for your questions and for I forgot your name. Siapa?
Aulia.
Aulia. Thank you for your nice presentation. Now let's move on to our next speaker, Ibu Irma Ratna Kartika.
Assalamualaikum warahmatullahi wabarakatuh. May the peace, mercy, and blessings of Allah be with you. Good day, everyone, and welcome to my presentation. First of all, let me thank you all for being here today. Let me start by saying a few words about my own background. My name is Irma Ratna Kartika from Chemistry Program, Faculty of Mathematics and Natural Science, Universitas Negeri Jakarta. I'm here today to talk to you about simulation of vitamin C derivatives as antioxidants using molecular docking. As we know, reactive oxygen species can be produced by endogenous and exogenous. Exogenous sources such as ultraviolet light, ionizing radiation, visible light, smoking and pollutions, heavy metals, environment toxics, chemical drugs, infection, chemotherapeutics, and so on. And from endogenous sources, during the oxidation reaction of metabolic pathways in mitochondria, endoplasmic, reticulum, peroxisomes, nucleus, plasma, membrane, and inflammation. All accumulation of cellular walls and unbalanced rows can lead to the formation of the oxidative stress. Its manifestation can affect or oxidize the cellular content such as cell membrane, phospholipid, lipid, protein, and DNA, which in turn causes many age-related diseases, particularly cancer, early aging, arthritis, neurodegenerative disease, and diabetes. The body has several enzymatic mechanisms to minimize locally induce damage and to protect against excessive production of free radicals. Antioxidants play a vital role in this defense mechanism. In healthy organisms, protection against the harmful effect of reactive oxygen species is achieved by maintaining a delicate balance between oxidant and antioxidants. The continuous production of free radicals in aerobic organisms must be equalized by a similar rate of antioxidant consumption. Enzymatic or non-enzymatic antioxidants are substances that prevent the formation of free radicals and seek and neutralize or repair the damage caused by them. Vitamin C. Vitamin C is one of enzymatic antioxidants. Once oxygen reacts with the lipid change, the change in dipole moment will cause the peroxyl radicals to float to the interface. In addition, vitamin E removes the peroxyl radicals. Ascorbat can recycle vitamin E, and enzymes then remove the damaged fatty acid and insert a new one, repairing the lipids. Vitamin C application is limited because its unstable chemical properties, which can be easily oxidized or destroyed by light, aerobic condition, or oxygen, high temperature, copper, humidity, alkali, pH more than 7, heavy metals, and enzymes. Hence, a variety of vitamin C derivatives have emerged in practical antioxidant application to increase its thermal and stability. And some publications have reported several promising biological activity of vitamin C derivatives like antioxidant, anti-tumors, antiviral, anti-cancer, and antibacterial, and so on. Meanwhile, the caprylic acid is a medium chain C8 monocarboxylic saturated fatty acid that is enriched in coconut oil and goat milk and is also used as antimicrobial agent in the food industry. Recent study, octanoic acid can inhibit the activity of hydroxymethylglutarate coenzyme I synthase or HMG coenzyme I synthase. This enzyme catalyzes the formation of HMG coenzyme, which is an intermediate in both cholesterol synthesis and catagenesis of the tumor cell. In addition, octanoic acid also disturbs cell cycle protein or cancer cell, leads to inhibition of cell cycle. Based on the background, we designed a combination of vitamin C and octanoic acid as vitamin C derivatives to evaluate their antioxidant potential in silico study using molecular docking. So the aim of the molecular docking simulation was to investigate the promising of design compounds to engage to receptor site in a way that would disturb the ROS production, leading to maintenance of redox homeostasis. Molecular docking has been an important tool for studies of receptor-ligand interaction in the inhibition of enzymes related to antioxidant activity. This technique showed clarification about the possible region of the receptor where the activity occurs, what amino acid residues are involved in the interaction, and what atoms are directly interacting with the ligand. The 3D structure of the enzyme were downloaded from the PDB site and proceed by the Autodoc for T2 software program. We use two enzymes, LO and NO, that respond for the production of reactive oxygen species during the metabolism. This is the flowchart of the method. So the protein receptor selection, we downloaded from structure downloaded from the PDB site. And meanwhile, ligand search from database modeling, and the structure of the ligand and the commercial drugs convert into Muffin Sketch program, and both preparation of both structure by Autodock 42 tools for docking and by the use of Windows terminal docking is performed. After that, interaction analysis of top 10 compounds is carried out by BioVia Discovery Studio. And selection of a best compound for antioxidant agent will, and further on synthesis a compound in lab. Okay, this is the protein. It is the two proteins that we use in this article is a lipoxygenase and NADPH oxidase. And we signed 6 compounds with combined vitamin C and octanoic acid as vitamin C derivatives. These compounds acted as ligands for docking receptors of lipogenase and NADPH oxidase, and the structures of designed ligands can be seen in this table. And we use two original native ligands, protocatechoic acid and adenosine 5-diphosphate, and 2 commercially drugs, Zileton and dextromethorphan-dex, were used as positive control. All drugs and designed compounds were converted to 2-dimensional and 3-dimensional structures using Muffin Sketch program from ChemAxon. And finally, the docking of model compounds with the enzyme were visualized using BioVia Discovery Studio. Okay. The data about validation protocols for molecular docking can be seen in this figure. MSID values were obtained less than 2, and this indicated good result according to the literature. This result proved that the protocols used can be applied in the molecular docking analysis between the receptors of the antioxidant activity and ligand. This slide shows Gibbs energy binding values generated from the molecular docking between compounds and two receptors. The binding free energies for the molecules evaluated at each receptor are shown in this figure. These values were used to classify the best poses obtained in the molecular docking analysis. The larger the peak, the lower the delta-G, and consequently, the more significant the interaction between the receptor and the ligands for the antioxidant ability. Molecular interaction of the compounds with the LO receptors can be seen in this slide. There is 10 amino acids residues of LO enzyme show interaction with compound 1. Only 2 amino acids residues form hydrogen bond interaction. The interaction of compound 1 with the LO receptor produce highest free energy than 7 and 8. A total 3 amino acid residues found in molecules 1, 7, and 8, which indicated the degree of correlation between molecules. The similarities correspondent concluded the ability potential antioxidant of 1. Molecular interaction of the compounds with NO receptors can be seen in this slide. 5 amino acids of NO enzyme shows interaction with 6. 3 amino acid residue shows interaction with 6. Compound 6 generated the highest free energy than 9 and 10. A total of 4 amino acid residues found in molecules 6, 9, and 10, which contributed to the binding stability as a good antioxidant of 6 in the NO receptors. Visualization molecular interaction, including hydrogen bonds and hydrophobic interaction, is clarified using BioVia Discovery Studio. From this slide, we can see compound 1 generates 2 hydrogen bonding, while in native and gel only 1. Also in this slide, potential NO-receptor ligand binding interaction of compounds with NO receptors. Compound 6 produces 3 hydrogen bondings, and native ligand also 3 hydrogen bondings, and commercial drugs only 1 hydrogen bondings. According to the molecular docking simulation, in conclusion, in LO receptor, compound 1 produces the highest free energy, then 7 and 8. The interaction between 2 amino acid residues of LO enzyme and 1 exhibit 2 hydrogen bond interaction. Compound 6 generated the highest free energy, then 9 and 10. There are 3 hydrogen bond interactions between amino acid residues of NO receptor and 6. The result add to the evidence supporting antioxidant activities of 1 and 6 in 2 receptors, LO and NO, respectively, via Roche degeneration. Further research is warranted into their effectiveness in the decrease of oxidative stress for soluble commercial antioxidants. And finally, the synthesis of 1 and 6 can be performed to justify the molecular docking. Finally, I wish to express my most sincere gratitude and appreciation to Faculty of Mathematics and Natural Science, Universitas Negeri Jakarta, for the financial support. I would also like to wish my deepest thanks to my team members for their assistance and help in excellent research and composing the article. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Nice presentation, Bu Irma. Is there any questions from audience? Yeah, I would like to ask one question
Yes, please.
to Irma. Is there any chance to make a combination between vitamin C and maybe a natural product compound to get new maybe new derivatives that can have good antioxidant activities?
Yeah, thank you for the question, Bu Vera. Yes, with molecular docking, we can predict especially the interaction between the compound and especially the design compound that we use. So the purpose of the design, the purpose of the molecular docking is to minimize the synthesis process, especially the time, the temperature, and chemicals. Yeah. So I think we can combine, or we can have a synthesis, or we can design the opportunity or the possibility compound, and with a molecular docking, we can do the process. So in the lab, we will have the optimum process to do the synthesis. Yeah.
Okay. Thank you very much, Bu Irma. Now I would like to move to. I would like to welcome to Professor Dr. Jalifah Latif. Thank you for coming this session.
Thank you very much. So should I share my screen now?
Before we have your presentation, maybe our committee will present your profile video first.
Thank you.
Okay, Professor Dr. Jalifah Latif is our invited speaker in this room. She is an associate professor in National University of Malaysia, UKM. She expert in organic chemistry, natural product chemistry, and molecular analysis, structure, and determination of chemical properties. She is also a founder member of MET International Society for Natural Products. So it is nice to have you as our invited speaker. Professor Jalifah, you have 30 minutes to deliver your presentation. Then we will have 15 minutes for discussion. So the time is yours.
Thank you very much. I'll share my screen now. Okay, can you see my screen now?
Yes.
Yes.
Oh, okay.
Okay.
So I'll just move. So it's moving, yeah? All right,
Yes.
okay. Okay, so Assalamualaikum warahmatullahi wabarakatuh dan selamat siang. First of all, I would like to apologize for the changes in the schedule because I didn't realize that I have to present at 1:30. I was looking at the previous schedule, which the invited session will be starting at 3:30 p.m. Indonesian time, which is 4:30 in Malaysia. So only that when I contacted my students that will be presenting later, and she told me that mine should be now. So I was like panicking and so on. And I also have a little bit trouble in terms of the my laptops as I'm just coming back from traveling. So maybe my laptop becoming haywire because been connecting to several Wi-Fi. I just back yesterday, Monday night. Yeah, I just back from the overseas trip on Monday night. So but anyway, we are here now. So I would like to share some of my research work that we have been doing post-pandemic era. So I will walk through with you on what we have been doing so far and what have we adjusting due to the affecting of COVID-19, yeah, post-pandemic of the COVID-19. Okay, so as been introduced, I'm Jalifah Latif. I work at the Department of Chemical Sciences, Faculty of Science and Technology, University Kebangsaan Malaysia, or known as the National University of Malaysia in Bangi. I'm a chemist by training. And my sub-specialization is on molecular structure elucidation, in particular using NMR spectroscopy and mass spectrometry. Okay, so my topics today, translating natural product research in post-pandemic era. So my presentation, I will walk you through, I will give a little bit view on COVID-19 pandemic, very short introduction, and I will walk you through on our research of our nature products, what are the advantage, opportunity and challenges, and how does pandemic affected our research, our research activity. And from that, subsequently, I will be showing you some example where we are translating our natural product research in the post-pandemic era. Okay, so let's look at what happened in 2020, where we are all over the world. We are facing this sudden COVID-19 pandemic, which started in the early March, 2020. Where along the way, if you look at the timeline here, along the way, we have been through several MCOs, movement control order. So in Malaysia, we have MCO, CMCO, EMCO. And there's I don't know how many CO. I can't really remember now, but it really does affected our research, our teaching and research activities in the university. So I would say, even though I would say sometime around August, the MCO has been enlightened a bit, has been some sort of like I think they allow us to go back to the university, but very limited access. Only selected people can be returned to the university. So I would say by end of 2020, there are zero research activities at, in UKM. And then once in 2021, we start to move back to the university, but unfortunately, most of the research still cannot be done because the labs and the instrument has been abandoned or has been stopped for quite some time. So it is quite difficult, especially on the instrumentation, where when the instrument was put into halt or off, they will face instrument breakdown and so on. So they are among the challenges that we are facing. In fact, we also we are facing our building has been categorized as sick building due to the closure. So we only have limited access to the certain area in our building. So it is a very challenging time for us and for our research student in Malaysia. I would say in the UKM, well, maybe in other universities, they are not so much affected. Okay, so you would see that what happened after pandemic, that our work have been shifted to home. So for scientists like me, the one that usually do wet lab research, it is very difficult for us. It is very unusual for us because we always do our research work back, all the wet labs in the labs, and we are always depending on the instruments. So there are some changes in the research topic. Some are switching to COVID-19 related research, and the intensity and the intensification of research has been reduced. Some research has been back to normal. After quite some time, some research has been back to normal, but there are some research area that come to halt. So it is quite difficult, but we are trying to adapt ourselves. So in terms of you will see that the resources also been affected due to the COVID-19 crisis, and collaboration as well. Where I would say in terms of collaboration, it is still going on, but everywhere research activity has been slowing down, or some of them has been stopped. So it makes that the collaboration is just becoming at in a passive mode, yeah, rather than in the active mode. I used to assist my collaborators overseas with helping them in terms of providing instrumentation analysis. They used to send me samples so that I can do the analysis here because we have the instrumentation facilities. But due to the COVID-19, everything has been stopped. And of course, our instrument also is not working post-COVID. So that's the effect of COVID-19. Yeah. Okay, so due to that, there are fundamental shift in terms of research, ethics, and peer review in the era of COVID-19 pandemic. So we have to do a lot of adaptation. We have to change our research methodology. We have to reframe the research design and so on. Yeah, but at the same time, we have to do some translational to our research, to the ongoing research, so that the research can be moving forward. Yeah. So same as well, I'm highlighting here in terms of publication, yeah, because as an academics, we are not just doing research, but we have to showcasing, or we have to publish our research output. So if you look at during pandemic, the COVID-19 related manuscript has been high time, especially sometime around May until July, yeah? And then it will slow down a bit. But the non-COVID submission is more or less the same, and sometimes in certain cases, there are very less publication focusing on non-COVID manuscript. I myself experienced a tough time from last year until now, where when we send our manuscript, it is quite difficult for us to get it acceptance. We keep on getting revision by revision, several times of revision. And after all that tedious revision, at the end of the day, you got your manuscript rejected. So this is also another challenges that we scientists facing, yeah, post-pandemic era. Okay, so now let me walk you through my research area, which is nature products. So what is nature products? Nature products are molecules that found in living organism, and it is classified into two classes, which is primary and secondary metabolites. So the primary metabolites are the natural compounds that produce through primary metabolism, which is a chemical processes that carry out by all organisms every day to survive and to reproduce. And they produce the compounds such as amino acid, nucleic acid, protein, lipids, and carbohydrate, which all these classes of compounds or classes of natural compounds show obvious cell function. We know the function of these all these compounds. They occur universal or they occur everywhere in nature. While so these are the example of primary metabolites. While for secondary metabolites, okay, so for secondary metabolites is produced by a chemical processes that are usually unique to the organism, so it is not universal. Certain organism will produce that kind of will undergo certain chemical processes and will produce a certain secondary metabolites, and these secondary metabolites we classify it as a natural products. So among the class of natural products, which also classify as secondary metabolites, are alkaloids, flavonoids, terpene, and so on. So if I could mapping it into a flow chart, natural product can be defined into two. Okay, if you are defining natural product generally, okay, you are defining a natural product as any natural compounds that produced by organisms. So this is the big natural product up here. But if you are talking about natural product and you are specifically focusing on the secondary metabolites, then the metabolite that unique to the organism, you are talking about compounds, natural compounds like alkaloids, terpenoids, flavonoids, phenylpropene, and so on. Okay. And the sources, we know the sources of natural product, it can be found in bacteria, it can be found in microorganism, in animal, in fungi, and also plants. Yeah. And although primary metabolite and secondary metabolites, their function is so much different, but they are interrelated between each other. So you will see the primary metabolites will be formed, and throughout the formation of primary metabolite, there are few natural compounds that form, which is called building blocks. So these building blocks is the precursor in the formation of secondary metabolites. Yeah. So among here, the building blocks are shikimic acid, amino acid, aliphatic amino acid, acetyl coenzyme A, and so on. So these are all building blocks that will be used by the organism to produce secondary metabolites. Yeah, or the one that I'm talking about here is the natural products. Okay, so then when we are talking about the secondary metabolite or natural products, these are the processes that been through in the formation of natural products. So we have shikimic acid pathway, we have acetate malonate pathway, and we have mevalonic acid pathway. Okay, so these are just another view of how secondary metabolites such as terpenes, steroid carotenoids, alkaloids, flavonoids has been formed through several metabolic pathways. Okay, so why natural products? Okay, so because why we study natural products? Because natural products give an enormous scaffold diversity. The structure is very complex and is very diverse. So with a larger sp3 carbon, with a higher molecular mass, and have a greater molecular rigidity. So all these characteristics can be contributed into several biological activity that can be several biological activity that can be performed or can be shown by secondary metabolites. Okay, so this is what I meant by structurally optimized by evolution to serve particular biological function. Yeah, so because they produce secondary metabolites, they produce a nice scaffold which can be which is how to say which is very what we call it a wider chemical space. Yeah, so it can fitted, yeah, it can be fitted into interaction with a certain with a certain active site like protein and so on. Okay, and we know that natural products always been used in traditional medicine, and it is by looking at the usage in traditional medicine, we can provide insight regarding the efficacy and efficiency safety of the natural product itself. Okay, so if we look at by looking at a combinatorial chemistry, yeah, if you look at as in when you define statistically the molecular space for natural products, so you will see these are the molecular space of natural product in comparison to the drugs, yeah, to the developed drugs. So you can see the chemical space of natural product is similar to the drugs, yeah, compared to the combinatorial compounds. So this shows that natural products are a very good candidate in terms of developing new drugs. Okay, so when we look at traditional medicine, where can these drugs come from? It can come from information from traditional medicine and also biodiversity, where there will be high probability in finding new drugs. Okay, so discovering new drugs or bioactive natural products, it can be either chemotaxonomy based, ethnomedicinal knowledge, or random sampling, yeah, or serendipity. Okay, so because some of natural products, they will have all these biological activities such as cytotoxic, anti-malaria, and anti-infective, and so on. Okay, so strategies in natural product research, yeah, usually we will choose what type of organism that we would like to study. Either is plants, animals, microorganism, and so on, and it has to follow by extraction methods, finding the natural products, okay, and testing the natural products in several types of assay. So if we look at the strategy, okay, when we correlate it with the effect on the pandemic, so it is the strategy in natural product research is purely wet lab based. Yeah, means you need the lab. You need the lab to do the research. So it really been affected by this pandemic COVID-19 issues. Yeah. Okay, and this is also how nature products get involved in the drug discovery cycle. So you will see here when the compounds or the nature product has been detected, it has to go through several pipelines, yeah, where you have primary assay, secondary assay, and then it will bring you to the lead compound. The structure activity relationship study has to be done, and structural characterization, the ligand protein complex has to be established, and then you will have the design, the chemical synthesis to make sure the source of the new drug candidate is enough, and it will go through the same process again because you are producing the synthetic version, yeah, compared to the original nature product version. Okay, and there are several techniques, analytical techniques or instrumentation that is needed in assisting nature product research, such as NMR, ultraviolet, infrared, X-ray. All these spectroscopy methods, yeah, is needed when you are doing nature product research. So as I relate it to the effect of the pandemic, so all this has been put into halt because you can't access, yeah, you can't access to the instrument due to the movement control order during COVID-19 pandemic. So it's really put us as a nature product researcher in halt. Yeah? We can't do much, yeah, during MCO. Yeah. Okay, so these are all the analytical techniques that support the nature product research. Okay, so why we study nature products? Yeah, because we know nature products has come into the world recognition. We knew about Tu Youyou, yeah, the lady that win from China that won Nobel Prize in Medicine in 2015, yeah, for the discovery of artemisinin. Yeah, artemisinin has been used in anti-malaria drugs. Yeah, so she shared the Nobel Prize winner with somebody that with another scientist, yeah, Campbell and Omura, which discover ivermectin. Then we also know about quinine. Okay, quinine is the one that been found a long time ago, which been used in treating anti-malaria infection. Yeah, and surprisingly, okay, from quinine they develop a synthetic drugs non-chloroquine, and during COVID-19, chloroquine has been the main focus, or has been in high time, where it was proposed to be, a quick remedy, a quick remedy in combating COVID-19 while waiting for the vaccine to be accessed. Yeah. So I'm just showing how important nature product it is. And of course, you know about Taxol that has been used now, still being used now in a modern medicine to combat cancer. And morphine, which has been used in modern medicine in operation as well, because it is as a powerful analgesic. But unfortunately, based on morphine also, new synthetic drugs, yeah, or club drugs has been developed, such as ecstasy, which mimic the natural products morphine. Yeah, so I'm highlighting some of the disadvantages, disadvantages of the nature products. Okay, I wouldn't call it disadvantages. It's maybe it is some drawbacks. Yeah, I would say. Yeah, or the side effects. Yeah, from the discovery of nature products. Okay, and you know about penicillin. I'm not going to talk so much on penicillin, and aspirin as well. Where does aspirin comes from? Okay, it comes from the nature product. So aspirin is a synthetic molecule that inspired by nature products. Okay, a part of that, nature products also can contribute to toxicity. One of the example are aristolochic acid, okay, which can be found in vegetables, in food, yeah, which cause nephropathy, okay, nephropathy, which is a side effect, yeah. So not all nature products are bring good effect, some of them can cause toxicity as well. So that is the reason why the research on natural product has to be continuously looked into, yeah, so that we can avoid this toxicity effect. Okay. Okay, and recently we are talking, when we are talking about natural products, there are also legislation issue, yeah, where now I think in Indonesia as well there are debates. I'm not sure about what is the principle back of the government of Indonesia principle in terms of accepting this medical marijuana. Okay, but in Malaysia, yeah, in Malaysia we are still it is still considered as illegal drugs, yeah, it's still considered as illegal drugs. So we are not allowed to plant cannabis, yeah, at this moment, but it is still being debated, yeah, it is still being debated at the parliament. Okay, because we know that the medical cannabis which produce low THC, okay, can be used, you know, or can be utilized in medicine, yeah, rather than the other variety that produce high THC that can give some psychoactive effect, yeah. Okay, and another issues of while we are looking at natural products is because there are a lot of resistancy, yeah, antibiotic resistancy, where microbes is becoming cleverer and cleverer compared to us, yeah. They adapt. They can adapt the condition where the current antibiotic is no longer affected to a certain microorganism. Okay. So these are the drawbacks of natural products, where, you know, sometimes when you want to produce the natural product in huge amount, so it is very difficult. It's not possible to culture the organism or to grow the organism in huge amount. And when you grow it, you know, it won't produce the natural products that it usually produce in the wild condition. Okay, so these are all the drawbacks when you study natural products or when you do natural product research. Okay, however, by having the state-of-art technology, yeah, especially high-end instrument like LC, high resolution MS, there are a few drawbacks can be overcome. So these are a few examples of developing some anti-infective drugs. These are also another examples, okay, where they are using another approach, yeah, in terms of finding new bioactive compounds or new natural products. Okay, in this case, they are using bioinformatic as well. And this, I think, they also use some bioinformatics and also do this. If I'm not mistaken, they will. Yeah, they use bioinformatics. There are one more example where they are using homologous or heterologous biosynthesis, something like that. Yeah. Okay. So, okay. So that's basically a little bit introduction of natural products, and now I will walk you through on how do we as a natural product chemist, as a natural product researcher, adapt ourselves, yeah, in our research. How do we adapt ourselves post-pandemic era? So what did I did? I was venturing into metabolomics, yeah, looking into metabolomics or using metabolomics approach in natural products in my natural product research, where I chose one of the plants which is known as Clinacanthus nutans, or in Malaysia it is known as belalai gajah. Okay. So we found that this plant can be grown easily, yeah, can be grown easily. So we want to see whether if it's been grown anywhere, whether it has been cultivated in a larger scale, medium scale, or smaller scale. Is there any differences in terms of the production of the bioactive natural products? So what we do, we study on the how we process. How we process our the samples, yeah, where we are using several different types of drying methods. Yeah, we use freeze dried, we use oven dried, and we have air dried as well. So we found out that by using FTIR, yeah, by using FTIR as a tool, we found out that you can see the differences in terms of freeze dried and oven-dried samples. Yeah, and on top of that, we proceed to the NMR analysis. So all this analysis has been done pre-COVID. Yeah, it has been done pre-COVID. So I have all the data. So this is what we see. We compared the different types of drying methods: air dried, freeze dried, and oven dried, and also extraction methods. Yeah, the extraction method that we use are either soaking or we use sonication method. Okay, and these are the information that we found out from the NMR analysis. Yeah, using metabolomic approach. So we found out that actually there are no significant difference between the extraction method. Either you are using soaking or sonication, except sonication is slightly quicker. Okay. But there are significant difference when you dry your samples. Okay, so you will see that the freeze-dry samples contains more flavonoids and sulfur glycosides, while the air-dried sample contains terpenoids. Contains more terpenoids. Yeah, oven-dried contains terpenoid and sulfur and sugars as well. Okay, so you can see if you are looking on the bioactive flavonoids, so the sample has to be processed by freeze drying method. Okay, and after that we proceed of using mass spectrometry, yeah, to monitor what type of secondary metabolite that contributed to the differences, yeah, contributed to these differences, and we found out that from the metabolomics and multivariate analysis, we managed to find out what type of compound that are responsible in contributing the differences between those extract, whether freeze-dried extract or oven-dried or air-dried extracts. Okay, and we also tested our extracts using anti-plasmodial assay, and we found out the freeze-dried extract is the one who gave the highest activity. So that is the reason why we are trying to fish out what type of natural products that contributed to this biological activity, and we found out it is a flavonoid glycoside that contributed to the activity. So these are all the result, the multivariate data result that brought us to the conclusion that the antiplasmodial activity in Clinarctus nutans is being contributed by flavonoid glycosides. Okay, so that these are the compounds of the flavonoid glycoside responsible for the anti-malarial activity. So I'm sharing this just to show you that we are a little bit lucky that we have done all the analysis, all the instrumentation analysis, all the lab work, wet lab work, yeah, pre-pandemic in 2019. But when COVID-19 approach in 2020, we have all the data. So we have been using the time, yeah, the lockdown time to process all the data and come up with these results. Yeah. So the second example, yeah, is we are trying to translate our nature product research during post-pandemic. So a part of looking at Clinarctus nutans, one of my project is on roselle which is known as Hibiscus subdarifa. So in Indonesia, you call it rosella. Yeah. Okay, so in what we have done in UKM is that we have our wild rosella, the one that has been brought, I think, from Middle East, yeah, to Malaysia. And what my research group has done is I have a plant. I have the a plant scientist that work on it. So the plant scientist is developing several mutant, yeah, the wild type, and from the wild type we developed 3 mutant of rosella, where we label our mutant as UKMR1, UKMR2, and UKMR3. So you can see UKMR3 is carries green color, where the genes that produces the anthocyanins, yeah, the one that responsible for the color, has been knocked down. Okay, so these are the work done by the plant scientist. It's not me. Yeah? I'm a chemist. Okay, so so these are just a comparison of, these are the one that been brought in Okay, and these are the wild type, and these are our mutant type. Yeah, and we have license, or we have patent it. Yeah, we have patent it as a plant variety of Hibiscus subdarifa. Okay, so what we did at university level, so we keep on cultivating our mutant type to make sure that their diversity, or or the genetic coding is still is still kept, yeah, in the mutant type. Okay, but it doesn't go so much, yeah. Okay, and we found out that the compounds that produced by Roselle, such as delphinidine 3-O-sambobioside, cyanidine 3-O-sambobioside, these are the two major anthocyanin produced by Roselle. They are very, very expensive, yeah, when it comes when they are being isolated as a single compound. Yeah, so it costs about 4,000 ringgit Malaysia for only 10 milligrams. Okay, so what we did is we tried to develop a HPLC method, so that whenever we cultivated our mutant UKM R1, R2, and R3, we can keep on tracking that the major compounds contains in this wild type is still there. Okay, so we already developed and validated our HPLC method in determination of the major compounds in Roselle. And at the same time also, we are trying to develop the method to preparing a dried Roselle extract, and also to prepare the potassium hydroxycitrate, because at that time we were looking at hydroxycitric acid, because hydroxycitric acid was claimed to have lowering lipid activity, lipid lowering activity. Okay, so this both method we have patented it. And these are also we are trying to come up with a product where we produce optimum amount of HCA in an extract. Okay, optimum amount of HCA in an extract, and it is compatible to the HCA that been produced by Garcinia cambogia. The advantage of having HCA from Roselle is because HCA is edible. Yeah, of course, Garcinia cambogia is edible as well. It can be eaten. Yeah, but it is too sour. It is too sour compared to Roselle. Roselle, you can still take it. Even the sourness of Roselle can be taken by us, but not Cambodia. Cambodia is too sour. So that is the advantage of Roselle, a HCA from Roselle. So in the beginning, after we develop the mutant, we are focusing on HCA. And then a part of that, we expand our research, in Roselle by looking on its effect on cardiovascular system. Okay, so we have done a lot of study on cardiovascular, like looking at heart damage, whether it can give protective effect. We published a few articles regarding to our research findings. We also look at the molecular mechanism, looking at the calcium regulatory channel and so on. Okay, we have looking at the myocardial infarction and so on. So there are a lot of animal study has been done on our Roselle extracts. Okay, so these are also another publication regarding on Roselle, on our Roselle extract. So at the end, we come out. We have enough evidence to show that Roselle have cardioprotective effect. Okay, so by having all these evidence, by having all this scientific evidence, as you know, animal study, you have to be in the lab. So during COVID-19, everything has been stopped. So what we did, by having all this evidence, post-pandemic era, we translating our Roselle research. What we do, we are approaching some of the companies, the companies that interested in cultivating Roselle. So what we did in terms what we did, we have tried our mutant roselle, which is UKMR2, and we plant it in the field. Yeah, so we do some medium scale plantation, and it was successful. Okay, so now I am in the position of analyzing the content of our UKMR2 roselle that is planted in the field, and monitoring the major natural products in roselle, whether the major natural product in roselle are still sustained when we cultivate our UKMR2 in the field. Okay, so these are the example of our translating our natural product research. Okay, another one is I'm also studying on the edible bird nest. Yeah, we know that edible bird nest is a source of sialic acid, which is a very high value chemical compounds. Okay, so these are also another approach, because we develop a method on how to quantify and how to determine and quantify sialic acid from edible bird nest. So we have been approached by having that experience, or by developing that method, we have been approached by the industry. Yeah, because the industry is worrying about the waste of the edible bird nest during processing. Yeah, when they collected the edible bird nest, they will do some washing, things like that. So there are some sialic acid that leaching out during the cleaning or washing processes. So this is where they have this residue or the waste of the edible bird nest after cleaning or processing. So they came and approached us and seeking our help and assistance in terms of recovering the sialic acid, yeah, from the waste of edible bird nest. Okay, so that this is also another another example of translating our natural product research, yeah, due post-pandemic era. Okay, so we have done a lot of chromatographic methods and we upscale it. We developed some enzymatic hydrolysis and optimization, and we also confirm the pure sialic acid and produces throughout this process by using all the instrumentation analysis such as NMR and FTIR. We even redo the biological activity just to make sure that the activity is still there, and also we conduct some toxicological study as well. Yeah, because this sialic acid comes from the waste, yeah, the waste of edible bird nest. So a toxicology study is necessary. Okay, so these are the result that I can share with you, where we have several different types of edible bird nest waste and several different processes that we have done on the waste, and these are the sialic acid detected in each of the waste that been given to us. Okay. All right, and on top of that, also we are also trying to adapt ourselves into what we call care in natural products research. Care is an acronym for a lot of things. So you can have it. It has been introduced by Professor Cordell. Okay, you can read his article regarding care in natural product communication. These are volume 19, yeah, page 1 to 9. So among the care that he has been mentioned is critical action to respect the environment and concern about restoring the environment. So due to that, we working with nature products, okay, we have developed a hybrid supercritical fluid extraction in UKM. Okay, so we have this instrument in UKM. So these are basically the schematic diagram of how CO2 extraction is in action. Okay, and we have tested on our local herbs. Yeah, so this is misai kucing, or in Indonesia it is called kumis kucing. Yeah, where we develop how we try to extract the chemical marker using conventional methods and compare it with using supercritical fluid extraction, yeah, or SFE. Okay, so these are the results can be you can check on the result by going in by looking into our manuscript here, or our article here. It has been published. Okay, we also published another one. We also tried on another herbal as well, which is Philanthus nuriri, okay, and Labisia pumila. Okay, and the recent one that we are looking at in terms of nature product are looking at this very special small molecule which is called Cavacrol. Yeah, Cavacrol is a monoterpenoid, why it is so special? It is a monoterpenoid that carries aromatic ring, yeah, in the structure. And our preliminary study, we found out Cavacrol has shown some protective effects, yeah, in combating cardiomyopathy. We know that when we most cancer patient, yeah, will be treated with chemotherapy drugs. Okay, so one of the types of chemotherapy drugs are doxorubicin, and the side effect of doxorubicin is it will cause cardiomyopathy. Okay, it will cause a side effect, a very bad side effect to the heart. And by studying the molecular mechanism, we found out, yeah, by using in silico, we found out Cavacrol, yeah, Cavacrol managed to stop or to interfere, yeah, to interfere in terms of mechanism that produce cardiotoxicity or cardiomyopathy by doxorubicin. Okay, so Cavacrol either can stop at this receptor here, okay, or in SMART or in or at the targeted genes here. Okay, so these are my ongoing projects. So but I'm not sharing any results yet because it is an ongoing project. At the moment now, we are looking at Cavacrol, and we are also making Cavacrol derivatives. So we are using nature products, and this nature product, this Cavacrol is inspired in making the derivatives, and we are going to test the derivative, which derivative will able to combat, will give us the best activity in combating cardiomyopathy or in reversing the cardiomyopathy induced by doxorubicin. So you can listen to the preliminary work on Cavacrol, which will be presented by my student, Ms. Rini Ratnosari, at the end of this session. So with that, I think I'll conclude my presentation. So as a take-home message, okay, so nature products, yeah, nature products is, I think, post-pandemic era, it is still reliable, or it is still applicable in drug discovery, and especially in drug repurposing. So one of the example that I've already highlighted is the anti-malaria drugs, yeah, the chloroquine, the one that really come into high time during COVID-19. And also take home message that not all natural compounds are necessarily safe. Yeah, some of they are valuables. Yeah, but you have to test the safety and toxicity of the natural compounds. So not the statement or the claim saying that all natural are safe is not really true. Okay. All right. So and as I said, we are going into this industrial, or fourth industrial revolution now. Instrumentation, or high-end instrumentation such as HPLC, NMR, high-end mass spectrometry, these are all very, very important instrumentation to support research, scientific research, especially nature products research in the era, post-pandemic era, and in the fourth industrial revolution era. So with that, I thank you very much for your kind attention.
Thank you very much for your very informative presentation, Professor Jalipa. I think even in the COVID-19 situation, your lab still productive because many publication released, and then many natural product is applicative and maybe scale up in scale up for industrial. So is there any questions from the audience for, okay. There is a question from Ibu Irma. As we know, when we obtain the extract from certain plant sample, it is biological activity sometimes is different when we isolated the single compound from the extract. How do you overcome that such obstacle?
Okay, all right. So thank you very much for the question. Yeah, I think that is the common drawbacks that we face, yeah, as a nature product research, because all this while we have been approaching or doing research in nature products with what we call reductionist approach. Yeah, reductionist approach is where you started with extracts and then you narrow down up to a single compound. So that is the reason why now metabolomics is in trend, because metabolomics, you are looking on a bird's eye view. You are looking at the bird's eye view of what contains inside your extract, what contains inside the plants. Yeah, some of them may be effective, some of them may be just being there without having any effect. So that is why I am adjusting I used to do a reductionist approach as well as a natural product chemist. Yeah, I did that before, and I'm still doing it. Yeah, but I will be very selective when I do natural product research by choosing reductionist approach. Maybe if you look at if you find a new organism, you can still go on reductionist approach, yeah, because you want to know, you want to have an idea first. But once you have the idea, I found out metabolomic approach is very, very useful, so that you won't be having the problem that you are facing, the one that you mentioned to me. So I would advise to you of using this tool, this metabolomic approach, so that you won't lose the biological activity. So I hope I answer your question.
Okay, thank you. Is there any questions from other participants? So I would like to ask you one question. Is there any change in research topic due to COVID-19 since our government stopped the research grants if the topic is not related to COVID-19? How about in Malaysia?
Yes, okay, thank you very much for the question. Yes, I agree with you. I wouldn't say it has been stopped totally, no, but the funding seems to be lessened. Yeah, the funding to be lessened. So they are trying to prioritize. They are trying to prioritize the research. So more attention has been given to COVID-19 related research. Yes, I agree with that. It happened. The same happened in Malaysia. However, the ongoing research that been carrying on at the moment is still continuing, but you will have lesser budget. So that's why you really have to adapt yourself, in doing so. You maybe can do the research that you have done, the ongoing research, but you have tried to adapt or modify it. Like in my case, when we can't go to the lab, we can't conduct the animal research, yeah. But we need to expand our cardiovascular research, because in my area, I work with this cardiovascular group, for quite a long time. We have a very strong group there. But when during pandemic, nothing can be done. So everything has been put into stop. So it is very difficult. So what do we do is like, I'm trying to venture in terms of in silico methods, yeah, where we are using molecular docking, computational studies, and so on, just to back up on our research. And then by having some preliminary data from that, we are applying for the grant. Yeah, we are applying for the grant. So the amount that you get is not as much as before, but at least it will keeps you ongoing. Yeah. Hope I answer your question.
Thank you very much. Yeah, thank you very much, Dr. Jalisa. Hopefully I can visit your laboratory again.
Yeah, you're most welcome. Yeah.
Yeah, maybe almost more than 10 years ago, I visit your laboratory with Prof.
Oh,
Diana.
okay. Okay. You're
Okay.
most welcome. Yeah. We are trying to get
Okay.
new instruments. So hopefully we can assist you with that. Yeah.
Okay.
Okay.
Before ending your session, maybe we can take picture together
Okay.
with all the participants. I hope all the participants can turn on the camera, please. Okay. 1, 2, 3. And the committee would like to present a certificate for Dr. Jali Palatu.
Thank
Okay,
you very much.
Professor Jalipa, we would like to thank for your presentation. Hopefully, we can meet again in other session.
Inshallah. Thank you very much.
So we will move to other presentation from Wida Rosa Kartika.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Wida Rosa Kartika from Chemistry Department, State University of Jakarta. I will present to you about two flavon derivative from the ethyl acetate extract of the wood of Nangkadak, Moraceae, and their antioxidant activities. The topic that I will going to explain is introduction, research method, result and discussion, conclusion, and reference. Nangkadak is a plant of Moraceae family and Artocarpus genus, with Latin name are Artocarpus heterophyllus. This plant was first crossed in 2000 at Mekarsari Fruit Garden. On a previous studies that shown the flavonoid compound, which is phenylated flavons, has been isolated from the root of Nangkadak and has potential as antioxidant. In this slide, there are several phytochemistry of genus Artocarpus, such as Norartocarpetin, Artocarpesin, Cudraflavonsi, are isolated by methanol extract of the hardwood Artocarpus heterophyllus, and then Artoindonesianins U and Artoindonesianins V are isolated by hardwood Artocarpus champedon. This is research method. First, the collected wood of Nangkadak from Mekarsari Fruit Garden is isolated by extraction, fractionation, and purification. And then the structure of the pure extract was elucidated by UVPs, ER, NMR, and HMBC correlation. And the last step, to test the antioxidant activity with DPPH method. The powder wood of Nangkadak was macerated with ethyl acetate and then evaporated the sample until the extract was thickened. After that, we separated the compound by vacuum liquid chromatography and radial chromatography. Then we got the structure with elucidation structure, which is spectroscopy. So this is the result. There are two compounds has been isolated by ethyl acetate of wood Nangkadak. The first compound is cyclomyrosin, and the second compound is Cudraflavonsi. But I just gonna explain the compound two, which is Cudraflavonsi. Okay, this is the structure elucidation of compound 2 using UV-Vis. As we can see, the spectra, there are 3 peaks and the maximum absorption for flavon derivative compound in 313 nanometer for band 1 and for band 2, 261.5 nanometer. And then for band 3, 205 nanometer. Okay, this is the FTIR spectrum of compound 2, shows absorption at the wave number 3352.06 for the hydroxyl group, and then 1650.25 for the carbonyl group, and then 1464 indicated the presence of methane group, and 1359 indicated the presence of methyl groups, and the last 1620 and 441 for carbon-carbon double bond aromatic. Okay, in the proton NMR spectrum shows a singlet signal at 13.42 ppm, which is characteristic of the hydroxyl group at carbon 5. And then 3 aromatic signal at appear as ABX system at 6.55, 6.50, and 7.17, indicated that a ring D substitute at carbon 4 accent and carbon 2 accent. And then there is a singlet signal at 6.35 ppm, indicate a ring A substitute at carbon 5 and carbon 6, carbon 5, 6, and 7. And then another proton NMR spectrum appear a singlet signal as methyl, four signal as methyl, indicating the presence of 4 methyl phenyls, and then 2 doublet signal at 3.08 and 3.35 ppm for 2 methylene groups, and 2 multiplet signal at 5.12 ppm and 5.28 ppm for the 2 methane groups. This signal indicate the presence of 2 isoprenyl groups in compound 2. Okay. This is the carbon NMR spectrum. In this structure elucidation shows that there are 25 carbons, including quaternary, methyl, methylene, methane carbon, and carbon of the carbonyl. As you can see, carbon of carboxyl signals at 183 ppm, and then there are 4 carbon of hydroxyl, and then 4 carbon of methyl, and 2 carbon of methane at 22 and 24.6 ppm. Okay. This is HMBC correlation. This structure was needed to confirm the 3D structure, as the previous elucidation method was consist of two-dimension structure explanation. HMBC correlation, this structure can reconstruct the 3D structure with correlation proton to Mechburg. For the example, the proton of carbon 3 accent are a correlation with 4 carbon neighbor, which is such as carbon 1 accent, carbon 2 accent, carbon 4 accent, and carbon 5 accent. So this is the structure of Kudrow-Lafont. As you can see, the structure are the same as the literature. On the bioassay, the first compound, which is cyclomorosin, has a weak antioxidant activity with IC50 116 ppm, while the second compound, which is Kudrow-Lafont C, was inactive with IC50 more than 200 ppm. So this is a conclusion. Two flavone derivative, cyclomorosin and Kudrow-Lafont, were isolated from the wood of Nangkadak. Compound 1 showed weak antioxidant activity with IC50 116 ppm, while compound 2 was inactive with IC50 more than 200 ppm. So this is reference that we use. Then yeah, thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Okay, there will be one question from Irfan Maulana. Please, Irfan.
Thank you for the attention. Thank you, Bu Vera. I want to ask a question for Widya. Why you use wood of Nangkadak in this article?
Okay, thank you for the question. I'm sorry I can't open camera because my camera was broke. Okay. We use Nangkadak because in Indonesia there's a lot of Moraceae family, especially Artocarpus genus, and Nangkadak is one of them. So we use Nangkadak to isolate the pure compound inside of the wood. And then after got the pure compound, we can see the bioactivity that could give a benefit to for human being. But in this case, the compound has a weak to inhibit the antioxidant activity. But maybe the compound can be strong to inhibit to another bioactivity. If it's work, the compound can be reproduced with synthesis and used to medicine, drugs, or anything else. Thank you.
Thank you, Vida, for your answer, and Irfan for your questions. Now let's move on to our next speaker is Eva Handayani.
Assalamualaikum warahmatullahi wabarakatuh. I'm Eva Handayani from Chemistry Study Program at State University of Jakarta. Now I will present an article entitled Potential Antioxidant Flavonoids from Ethyl Acetate Fraction of Artocarpus champerinstem. In this presentation, there are 4 topics that will be discussed. First is introduction, the second is research method, the third is results and discussion, and the last is conclusion. For the introduction, Artocarpus champedon grows as a tall tree, branched with wide sap all over the plant. This plant has properties to treat various diseases such as fever, diarrhea, and malaria. It was reported in previous research that derivative flavonoids from this species exhibited significant bioactivities. The plant's material that used for this research is the stem of Artocarpus champedon that were collected from Taman Buah Mekarsari. The method that used for this research is extraction and isolation. The compounds that have been isolated were tested for their antioxidant activity. The sample of the powdered stem of Artocarpus champedon was macerated with ethyl acetate. Ethyl acetate extract was fractioned using vacuum liquid chromatography into 9 major fractions. The F fraction was separated using vacuum liquid chromatography and radial chromatography. The F10 to F12 fractions was obtained as compound artocarpanon. In this research, there are 3 compounds that were isolated. There is artocarpanon, artocarpin, and cudraflavon C. But now I only explain one compound, that is artocarpanon. Artocarpanon was obtained as a white solid. The UV spectra of artocarpanon displayed maximum absorption at a wavelength of 345 nanometer, 286 nanometer, and 211 nanometer, typical for flavonol derivative compounds. This is the proton NMR spectrum of artocarpanon. From the proton NMR spectrum of artocarpanon showed a characteristic signals of flavonol. A single proton signal at chemical shift at 12.16 ppm for a hydroxyl group at C5, 3 doublet signals in the aliphatic region, 3 aromatic signals that appeared as ABX system at C5 accent that were metacoupled to C3 accent and orthocoupled to C6 accent with chemical shift 3.84. Another aromatic signal appeared at chemical shift 6.03 ppm at C8. A single signal at chemical shift at 3.84 ppm indicating the position of O-methyl group at C7. Based on the proton NMR data and comparison of the spectroscopic data of this compound to that reported in the literature, compound was identified as artocarpanon. For the antioxidant activity, artocarpanon compound has a highest antioxidant activity with IC50 values of 99.31, artocarpin with IC values of 45, and Kudra Flaponsi with IC50 values of 17.724. The conclusion is 3 flavonoid derivative compounds, there is artocarpanon, artocarpine, and Kudra-flavon C were isolated from the stem of Artocarpus champedon. All compounds exhibited high antioxidant activity against TBPH with IC50 values of 99.3, 45, and 17.724 ppm respectively. This is the references. And thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Okay, maybe Gladys will ask for Miss Eva.
Thank you, Eva and team, for the good presentation. And I want to ask a question. In this research, you are here to determine the antioxidant compounds using IC50. What is the IC50 value range of antioxidant compounds? Thank you.
Okay, thank you, Gladys, for your question. About this question, a compound as an antioxidant is very strong if the IC50 value less than 50, and if the IC50 value strong, it's from 51 to 100, and if the IC50 value in moderate is from 101 to 150. Thank you.
Okay, are there any questions? Okay, we will let's do the next presenter, Mr. Dwi Atmanto. I will share the video.
Assalamualaikum warahmatullahi wabarakatuh. I will introduce myself. My name Dwi Atmanto, a lecturer from Faculty of Engineering, Universitas Negeri Jakarta. I will present my research about the social capital for riverbank management. The title of my research is a model of river environmental management based on sustainable community social capital. The Ciliwung River, one of the vital rivers in the nation capital, is also strategic because about of the river is used by the people who live around the river banks. The Ciliwung River has many problems, including causing flooding, clean water rises, sedimentation, construction of buildings along the river body due to population growth, damage to forest land into settlements, land changes in upstream areas, disposal of domestic waste to industry causing pollution. Various government approaches to overcome the damage to the Ciliwung River from river naturalization program to river revitalization have not been optimally implemented. Communities have an important role in preserving river banks because communities along river banks also have energy that can be empowered. Social capital owned by local communities is a driving agent and can be mobilized in accordance with the principle of togetherness, mutual trust, mutual benefit for mutual progress, and equality of ideas. Various government efforts to overcome the damage to the Ciliwung River from river naturalization to river revitalization have not been optimally implemented. Communities have an important role in preserving river banks because communities along river banks also have energy that can be empowered. Social capital owned by local communities is a driving agent and can be mobilized in accordance with the principles of togetherness, mutual trust, mutual benefit for mutual progress, and equal of ideas. The settlement of problem which is discussed in this paper includes how are some social capital and cultural strategies that are run by riverbank communities in response to regular flood and the uncertainty of riverbank development policy. The method of this research is by survey with qualitative and quantitative description, and next to continue by a dynamic system. The study used a descriptive method that gives a real picture. The data was obtained through a survey of 30 people living along the river banks and community organizations that care about river damage in the Depok area. Data were collected through interviews, observation, and filling out questionnaires. The data collected were analyzed qualitatively and quantitatively. The research took place in the Depok area of West Java, covering the district of Sukmajaya, Pancoranas, and Beji. Ciliwung River Care Community includes Ciliwung Friends, Ciliwung Caring Community, Ciliwung Caring Student Community, and then NGO. This is the research location in the Depok area. And the data from the questionnaire to tabulate and to get the table about the relationship of potential social capital with participation in managing rivers. Like this, and the description about the table. Next. Based on the table shows that the social capital response to the participatory aspect is high at the 60.17%. The medium reciprocity information exchange activity is 39.09%. Aspect of trust, mutual trust and respect, which is high 63.48%. Aspect of norms, a set of customary rules or local rules that are enforced, 97.6% are high. And aspect of values is the ideas of community ideas are 64.20% high. And the table data next to analysis by statistic were carried showing F count more than F tabel is 14.45 more than the 3.372. This shows that the percentage composition in each aspect of social capital is significantly different, where the implementation of the norm aspect occupies the position of 97.6%. And then the social capital model in this picture is showed that the mutual trust, mutual respect, mutual benefit is the triangle, and the side of this triangle is cooperation and solidarity, network and scaling up, and then the bottom of the triangle is competitiveness and sustainability. Social capital is each community, group, and region is different. There are areas that have strong social capital and vice versa. This is the influence by the characteristic of the settlement and the cultural nature of the local community. And this is the scheme of the picture social capital, which to endorse the participation level, implementation of the reforestation along the river, implementation of the clean list of the river environment, river monitoring, and acceleration of the river flow, and next establishment of the upstream and downstream waste management. And all this to go, and next to get a sustainable river management. Community social capital cannot stand alone. Community groups must establish cooperation with government agencies, the private sector, and universities. Universities have knowledge and technology that can strengthen social capital through programs, materials, and technology. And this is the model implementation, social capital for river management and social capital to support by the local government and community, and to get the river bank management by technology and knowledge and management from the university and then college and from the government. And this scheme, the figure, to get the management model, dynamic system, and the result of dynamic system, you can see the result like this. And dynamic system model type 1, if the community participation without being based on the principle of social capital in managing river, water quality is easy to stop. Water quality initially increases, but then it decreases as the community's attention is weak. And this is the type 2, if community participation is river water quality management with less optimal government, support leads to improve river water quality in a long time. And this is, we can see that the activities of participation from the community, and but not to support by our government, and with the technology and the knowledge and the quality of river water quality management is can be to go to improve, but in the long time. There is a sense of trust between each other, help each of when there are members of the community who are not working familiar and tolerant of others. The social order of the residents that has been nurtured for a long time has produced a result in the form of caring among fellow citizens, shown by all heads of families working even in the informal sector. The existence of a social network and social network that participate active in every activity needed by citizen maintain social relation. And with the essence of the strong social capital in the culture community and supported by government programs, a river bank ecosystem can be realized that reduces river damage. Conclusion, community groups who care about the condition of the Ciliwung River Bank have used social capital to manage the river. The aspect of social capital shows participation is implemented quite high, 66%, reciprocity 39.09%, trust 63.48%, norms 97%, and values 64.20%. The result of the dynamic system simulation show that the application of social capital by the community is able to improve river quality even though it takes a long time. Thank you for your attention.
Thank you, Pak Dwi Atmanto. Maybe we can continue to your next presentation before the question and answer questions. Okay, let's move to next topic.
Assalamualaikum warahmatullahi wabarakatuh. Introduce myself. My name is Dwi Atmanto from Faculty of Engineering Universitas Negeri Jakarta. I will present about the function of the mangrove due to the cosmetic, especially hand soap. Mangrove leaf extract substitution of formulation in making hand soap to improve environmental health of coastal communities Muara Gembong District, Bekasi Regency, West Java. This is the title of my research. And for the introduction, the Indonesia archipelago has the largest mangrove forest area in Asia. It is estimated that there are 22 types of the mangroves growing in Indonesia from 44 species in the world. The amount of the mangrove plant waste such as branches, leaves, and fruit is scattered and becomes useless waste. Many mangrove plants grow in the coastal area of Muara Gembong District, Bekasi Regency. Mangrove plants are widely known to as antiviral, antibacterial, and anti-ulcer and inflammatory. Mangrove leaves contain minerals such as sodium, potassium, calcium, steroid or triterpenoid compounds, saponins, flavonoids, and tannins. Due to the presence of these metabolites, mangrove leaves cause mangrove leaves to be used as medicine by the community. Phenolic compounds contained in mangrove leaves antibacterial properties. Tannins act as protein, while flavonoid work antimicrobial, and antiviral. Mangrove leaves can be used as the antibacterial ingredient in the manufacture of hand soap and other sanitizers. The dominant type of mangrove plant on the coast of Muara Gembong is Rhizophora mucronata. Mangrove Rhizophora mucronata has the highest carbon content among other mangrove species, which is 17.60 tons per hectare. Hand soap and other sanitizer are urgently needed for self-care to prevent the spread of the COVID-19 outbreak. This is still happening in Muara Gembong and other regions in Indonesia. The risk method was carried out by experiment. The tools used are measuring cups, beakers, funnels, stirrers, scales, packaging bottles. The material used are mangrove leaf extract, Rhizophora mucronata, 126 grams of Texapon, sodium chloride as salt, 125 grams of the immal, sodium lauryl sulfate, and then 10 grams of sodium chloride, amphitol 10 milliliters of fragrance, there 5 milliliter, and 2.5 liters of distilled water or Aquades. This is the preparation formula for making hand soap with the different mangrove leaf extract. Preparation formula for making hand soap with the F1, F2, F3, and F4 as formula. This is the Texapon ml soap, sodium chloride, amphitol, fragrance dye, Aquades is the same formula, but mangrove leaf extract with the different composition. F1 0.005%, and next in the formula is the 0.025%. The product quality test is organic test, pH test, and foam test. Organoleptic tests were carried out by 15 coastal communities in the Muara Gembong area, Bekasi Regency, which is overgrown with the mangrove forest. The data collected were analyzed by the F-test statistical to see the difference in the scores for each group of formulas. Panelists from coastal communities Muara Gembong, Bekasi, there are 15 people as panelists, as consumers. And the mangrove leaf extraction, the method of extraction mangrove leaves, Rhizophora mucronata, using the maceration technique was carried out for 20 hours by the take for 50 grams of the fresh mangrove leaves and cut them into small pieces. Enter the mangrove leaves that have been cut into 150 milliliter of Aquades. Mash the mixture of the mangrove leaves and Aquades using a blender. This is the extraction process. And the result of this discussion in here is product with the different formula. We can see there are 4 product of the hand soap, A, B, C, D, and the process of the hand soap. Based on the table above, it is and the hand soap mangrove leaf extract is the most preferred by the panelists, achieving an average score of 4.26. FCON shows FCON 3.299 more than the F-table. 2.272 shows that is a significant difference in the treatment for organoleptic tests. And this is the result of the organoleptic parameter of hand soap and organoleptic parameters to get a form, color, scan, texture, and this is the average and category, which is the F1 normal, F2 normal, F3 light, and 4 like. This is the from the answer plus panelists. And for pH test, the average pH of the hand soap and the average of the pH is more than 8 score pH. This is assert that the hand soap is the best. The table shows that the pH value for its hand soap is different. The highest pH value in the F1 group was 9.27. F-test shows that FCON more than F-table. This indicates that there is a significant difference in the pH value of the treatment given. For hand soap foaming, the test result, the difference of this preparation hand soap is more than 6.0. But in the here 76, 78, 80, and 79. But in the if we can see the formula 3 in here is the highest. They are 80.42. Based on the table, the highest mean is in the formula 3, which is 80.42 millimeters, while the foam power of the formula group shows that the foam power number is 76.01. The result of the statistical test of foam power showed that the FCON is less from the F-table, which that means there was no significant difference in foam power in its group of formula and soap. Based on the above experiment, or pH value above 8 indicates that hand soap given mangrove leaf extract is alkaline and still safe according to hand soap production standards. Formula 3, where the addition of 0.02% mangrove leaf extract was the panelist's highest reference for the coastal community of Muara Gembong. And then the average foam level of the preparation is above 78 millimeters. For the conclusion, that the mangrove leaf extract contains phenol and flavonoid compounds can be used as a substitution as a bacteria and glycerin ingredients for the making hand soap. It can be used in making hand soap, although hand soap liquid will turn slightly brown due to the oxidation with air. On some communities who live on the coast of Muara Gembong sub-district like hand soap products with the addition of 0.02% mangrove leaf extract based on form, texture, forming, skin, and color. Thank you very much. This is the presentation of the addition mangrove extract to making hand soap. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Pak Dwi Atmanto, for your presentation. Is there any question from other participants or Pak Dwi Atmanto? Okay, no questions for Pak Dwi Atmanto. Thank you very much, Pak Dwi Atmanto, for your presentation. Now we move to the next presenter. Okay, we move to Maharani Anska Azahra.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. I'm Irfan Maulana and my partner Maharani Anska Zahra from Salmonella Wenje team. We are the author of paper Optimization FNBA gene target for potential detection Staphylococcus aureus as foodborne pathogen bacteria using polymerase chain reaction method. This research is funded by PUI UNJ with contract agreement number as shown. First of all, allow us to warmly thank the attendees of this important conference for giving us the privilege of welcoming and addressing you all. For us, it's an honor and pleasure. Next, we will begin the presentation. Foodborne disease can happen in a wide range of areas. 18,462 cases of food poisoning occurred in the world as of 2020. Staphylococcus aureus is one of the pathogens that can cause a broad spectrum of infection. The optimization for Staphylococcus aureus detection need to be developed to produce more accurate and relevant results with PCR method. Next. Food poisoning case in Jombang caused by Staphylococcus aureus bacteria infected 105 people. Staphylococcus aureus can cause infection from superficial skin, infection to severe, and potentially fatal invasive disease. Staphylococcus aureus does not form spores. Staphylococcus aureus can grow in a wide range of temperature. Staphylococcus aureus found in milk, beef, and bread products. Staphylococcus aureus producing toxins staphylococcal enterotoxin. Next, the methodology. First, we were doing a primer design. The second, Staphylococcus aureus will culture on selective medium mannitol salt agar at 37 degrees Celsius for 18-24 hours. We were then inoculated in liquid media brain herd infusion at 37 degrees Celsius with aeration shaker at 150 rpm for 18 hours. Then isolation of bacterial genomic DNA using a commercial kit thermoscientific. The purity of the isolation result was measured using a nanodrop. After that, was carried to find the optimum annealing temperature primer for FNBA gene using gradient PCR. The amplification result were electrophoresis using 2% agarose gel and visualized by a UV transluminator lamp. For the next slide, my partner Zahra will continue the presentation.
Thank you, Irfan, for your presentation. Next is primer design. The FNBA gene size is 321 base pair, is in the order of 2535383 until 2538511 base pair. And the primary target FNBA genes for Staphylococcus aureus have an amplicon in 187 base pair. The FNBA gene used as a fluorescence factor present is abundant in aureus bacteria, so that we choose FNBA gene as target for detection as aureus bacteria. And then for the FNBA gene, and for the FNBA gene, as fibronectin binding protein A function for S. aureus is to infect its host to make it easier. And then the table shown is for Staphylococcus aureus gene FNBA primers for forward and reverse primers. Then for the next slide, results and the discussions is for growth of S. aureus. On the left picture is Staphylococcus aureus bacteria on mantle salt agar produce yellow colonies, and then we use brain heart infusion broth results in a color change, which indicate and then for the next step is DNA isolation. Here are the results for the DNA isolation for the quantification or the qualification results for the DNA bacteria DNA. In the range of 1.8 until 2 indicate the result of pure DNA. So the results for the DNA isolation is in 1.843, and so the results of the isolation of bacteria cultures of S. aureus or can be used as a PCR templates. For the whole genome, S. aureus size at HHA 25923 size is 27.5 kb, and here on the right picture is a qualification for detection. Here's in the A is letter and B and C as the DNA genome of S. aureus. For the results of the electrophoresis of DNA isolation shown that the size for the DNA isolation is matched with more than 10,000 base pair size. And then for the characterization of the central electrophoresis, the gradient PCR stage first is initial denaturation of 9695 degrees Celsius per 2 minutes, and then denaturation of 95 degrees Celsius for 30 seconds, and the annealing of use range from 53 degrees Celsius until 22 degrees Celsius. And then the results shown that the amplification band of FNBA gene as targeted is matched with 187 base pair. For the conclusions of the study is the optimization of FNBA gene target for potential detection of Staphylococcus aureus using PCR method have been successfully carried out. The primary design of FNBA gene can successfully amplify targeted FNBA gene on Staphylococcus aureus. The optimization using PCR method amplification results in an annealing temperature of 60 degrees Celsius, shown that still can produce targeted anticoal size 187 BPa. So for the next stage, the optimal annealing temperature of an FNBA gene primers can be further used to develop detection kit for potential detection of S. aureus in food using real-time PCR. That's all from us. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Is there any question from other participants? For Maharani and Ivan? Okay, there is no question. So we move to the next speaker. Is Chika Safa already here? Okay. So we can have your presentation right now.
Assalamualaikum warahmatullahi wabarakatuh. Hello, all of honorable judges. First of all, I want to say thank you very much for the opportunity that has been given to me. Let me introduce myself. Hello, my name is Chika Safa Maura. I come from chemistry study program at the State University of Jakarta. I will present about antibacterial and physical properties of kerosene, Muntingia calabura liquid source from bulk cooking oil. In line with population growth, increase in food is increasing. This is in line with the increasing amount of food waste. Cooking oil is one of the basic needs of the Indonesian people. Commonly known, Indonesians really like to eat greasy food. Cooking oil consumption in Indonesia reached 290 million tons per year. Poor management of used cooking oil can cause various negative impacts. The impact of the waste if discharged into the soil will reduce soil fertility and can affect the mineral content in clean water. When disposed of in aquatic environment, it has implications for the destruction of aquatic ecosystem, and if it is discharged carelessly in waterway, it can cause food pipes. From the health side, also, when used cooking oil is consumed, it can cause health problems. High consumption of saturated fatty acid result in elevated cholesterol level in the blood. This can cause burst of blood vessels and result in heart disease. Consumption of used cooking oil can also result in oxidative damage. Therefore, a breakout is needed to take advantage of used cooking oil, one of which is by making it an ingredient in hand soap. This potential rationally applied because used cooking oil contains fatty acid that can be made into subtract a saponification reaction. Nowadays, public attention to body hygiene is increasing, especially hand hygiene. This is inseparable from the effect of the COVID-19 virus pandemic that is still hitting Indonesia. One of the measures to prevent the spread of the virus is to wash your hands with soap. In an effort to increase the effectiveness of hand soap, active ingredients are needed that are able to kill virus and bacteria quickly. It is known that kerosene leaf contain antibacterial active ingredients that are flavonoid, tannin, and saponins. The method used an experimental method with concentration variations. The surface test for antibacterial activity using the distribution method and physical parameters for a free alkali moisture content from Hague and pH test. Result and discussion. First test, free alkali test. Basically, the free alkali test determines free alkali by neutralizing the base using titration of determine how much alkali should be neutralized with HCl. The volume of HCl in titration is obtained in the following table. Based on this formula, the result of the free alkali test value are obtained in the following table too. Based on the result in the table, it is shown the average percentage of free alkali levels in Kersen soft samples is below 1.4% or SNE standards, and various sample concentration where these levels are standardized, the feasibility of a soap that safe to use for the skin. The result of the sample are a reference that the sample will not irritate the skin due to the alkaline content. Second test is moisture content test. The water content test is carried out to determine the percentage of water content in liquid soap. The result of the moisture content test can be seen in the table. Obtain the result of moisture content as follows. According to SNI, the moisture content in liquid soap preparation is a maximum of 60%. For the result obtained, all concentration meet the standards set by SNI. Third test is form height and form stability test. Form height test aims to see the form power of liquid soap. The result of the height of the form after shaking for 20 seconds can be seen in the following table. The result of form stability can be seen in the following table too. Form stability after 5 minutes of form should be able to withstand between 60 until 70% of the initial volume. The form on the soap serves to remove oil or fat on the skin. If the form processed by soap is too high, then it can make the dry skin. When the fat on the skin is lost, it will make the skin more susceptible to irritation because the fat on the skin is useful as a defense. The uppermost layer of the skin is called the skin barrier. One of the constituents of skin barrier is fat. Fat will make the skin barrier tighter so that bacteria and microorganisms are not easy to enter the body. The difference in form height between formulas is due to the content of saponin compounds in the formula. Formulas with a high ethanol extract content cause indirectly to contain more saponin so that the form that is cursed is higher. In addition, the form stability is getting better. The form will last a long time, which eventually decreases. However, the data obtained are not in accordance with the theory. This can be due to difference in strength during manual shaking of each concentration. This can affect the high yield of form obtained mixtures containing the active ingredients of soap will produce a stable form when mixed with water. The fourth test is pH test. The soap acidity testing or soap pH test is carried out using a digital pH meter that has been calibrated with a pH buffer solution of 10. In the pH test, the soap is repeated 4 times. Calibration of tools and repetition of pH measurements aim to make the data obtained accurate and minimize the value of measurement errors. The pH value of soap can be seen in the table. Table shown that the addition of ethanol extract of kerosene leaf affect the pH value. The more addition of ethanol extract of kerosene leaf, the pH of soap decreases. This happens because ethanol is an alkaline atmosphere will act as a weak acid. Ethanol has the ability to act as an acid due to its ability to ensure proton from its hydroxyl group. The pH test of soap is carried out to ensure that the soap used is safe when used on the skin. The acidity of hand washing liquid soap must meet Indonesian national standards or SNI, namely pH 8 until 11. Formulas 1, 2, and 3 don't meet the SNI, while formulas 4, 5, 6, and 7 meet SNI. And the last test is antibacterial test. Test the antibacterial effectiveness of liquid soap of Carson leaf ethanol extract on Staphylococcus aureus and Escherichia coli bacteria using the diffusion method. This method is used due to the simplicity of the technique and shortness. Besides, it is often used for antibiotic sensitivity testing. The result obtained from the antibacterial effectiveness test of liquid soap of Carson leaf extract through observation for 1 day, with an incubation period of 24 hours, and with 3 times the rule for each liquid soap preparation can be seen in the following table. The left table is result of antibacterial test of soap against Staphylococcus aureus bacteria, and the right test is result of antibacterial test of soap against Escherichia coli bacteria. The result of the antibacterial effectiveness test of liquid soap of Carson leaf ethanol extract at a concentration of 5% are categorized as optimum against Staphylococcus aureus bacteria, and at a concentration of 3% categorized as optimal against Escherichia coli bacteria. From the result of this study, result of antibacterial effectiveness with a concentration of 3%, 5%, 7%, 9%, and 11%, Carson leaf extract perfect, a greater inhibition zone than without the addition of Carson leaf extract or concentration 0%. So it has activity good antibacterial. Conclusion, the soap from bulk cooking oil add with ethanol extract of Carson has antibacterial activity and good physical properties. That's enough for me. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Chika, for your nice presentation. Is there any questions from audience? So maybe I would like to ask you one question. If I'm not mistaken, Carson we can isolate metabolic metabolites from Carson, which has maybe different activities, different bioactivities such as anti-diabetic. So, is there any information about Carson which can have other activities, or maybe can have other bioactivities from metabolites from Carson?
Permission to answer, Bu Vera. The extra Carson contain alkaloid, flavonoid, and tannin, and there is activity antibiotic.
It seems Chika have a internet problem. Okay. Maybe we can continue to other speaker. We have Gladys Indira Putri will deliver the presentation.
Hello, serious global problem for the spread of pathogen and microbes. One of the microbe food poisoning is Yersinia enterocoltica is a gram-negative and facultative anaerobic. Yersinia found in poultry, egg, lamb, beef, and pork. Yersinia and yersiniosis that have simple nausea, vomiting, body diarrhea, and joint pain. To determine the underlying temperature, YMOA gene primer used. After obtaining the range temperature, it can be followed of the Yersinia and YMOA gene using the real-time action method. This is the main our research. We divide it in major steps. First is primary preparation of culture sample. Then and the last is optimization.
So sorry, we have to stop this video. I think the sounds of the video is not clear, so we have to replay the video. Thank you.
Isolation, and the last is optimization of underlying temperature. This is the gene that we use for this research as a target for PCR process. The YMOA gene is a virulent gene in Yersinia enterocoltica. The function is to modulate the gene expression by influencing the DNA topology. Topology DNA is talking about the property and geometry of DNA. The way they work is to make a break into the DNA strand and then pass the DNA strand and reclose the strands. DNA can be broken because the long DNA is very flexible. This is the nucleotide sequence of the YMOA gene from Yersinia enterocoltica in size of 375 base pairs of the complete genome. This is the primer that we use in our research. The primers produce the amplicon size at 185 base pairs. Based on in silico analysis, we obtained that the parameter of gene for GC content is 50% for forward primers and 45% for reverse primers. The primer length both of forward primers and reverse primers are 20 base pairs. This is the bacterial culture of Yersinia enterocoltica ATCC 23715. The picture on the left slide indicating the appearance of Yersinia enterocoltica colony with the small gray color on blood agar media. The process of growing Yersinia enterocoltica bacteria in blood agar media, we use strict plate method, and then we incubate at 37 degrees Celsius for 24 hours or overnight culture. After that, two colonies that grow in blood agar media were taken using a loop, and then grown in tritic soy broth media and incubate at 37 degrees Celsius for 80 hours or overnight culture at aeration shaker 150 RPM. The turbidity formed on the tritic soy broth media indicated that Yersinia enterocolitica successful growth. The quantitative data that we obtained from the isolate is 70.5 nanogram per microliter for concentration, and the purity is 1.88. We use spectrophotometer nanodrop to measuring the concentration and the purity of DNA Yersinia enterocolitica. And for the qualitative data, we check our isolate on agarose electrophoresis agar. We can see the band of Yersinia enterocolitica appear at above marker, make in the lane through 2 and 3 duplo as a duplo. This data shown that the isolate of Yersinia enterocolitica have the size bigger than 10,000 base pairs. And the last, we do the optimization and temperature of YMO agent primer pairs. We can see the primer can amplify at 185 base pair at all the temperature that we analyze. And for the optimum temperature, we obtain at 53 until 62 degrees Celsius. Based on the bright and only one band appear on the electrophoresis, but based on our previous research, the 60 degrees Celsius as an optimum temperature because this also the default annealing temperature for PCR method. So we choose the 60 degrees Celsius for next research at real-time polymerase chain reaction. For positive control, we use FIMC Salmonella TP at can be amplified at 95 base pairs. Conclusion that we obtain at this research: first, the polymerase chain reaction or PCR method was successful in optimizing the primer annealing temperature of the YMO agent for Yersinia enterocolitica. And then the result of the primer design of YMO agent that have been confirmed can amplify the DNA fragment of Yersinia enterocolitica ATCC 23715 with an amplicon length of 185 base pairs at an optimum annealing temperature of 60 degrees Celsius. Thank you for your attention. And also thank you to Kemendikbudristek for founded this research. Bye-bye.
Thank you so much for your presentation, ladies. Is there any question from other participants? Okay, there's one question from Vida.
Okay, thank you for the good presentation, ladies. I want to ask, you say that the tryptic soy broth have turbidity. How to know the turbidity in the TSP, the liquid media? Thank you, Gladys.
Thank you, Ida, for the question. To know the turbidity in tryptic soy broth media, we use optical density 600 to determine the turbidity, and we got the value is 1.6. Based on previous research, the good growth of bacteria is shown when the optical density is above 1. So the turbidity indicated Yersinia enterocoltica successful growth. Thank you.
Okay, thank you, Gladys.
Okay,
Thank you,
is there any questions? If there is no questions, thank you, Gladys, for your presentation. Now we move to our last speaker in this room, Ibu Rini Retnasari.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Firstly, I would like to say thank you to the Committee of Science and Mathematics International Conference 2022 for arranging this event. A little bit about myself. My name is Rini Retnosari. I'm a student from University Kebangsaan Malaysia, supervised by Dr. Jalifa Latif. Today, I will present my research entitled Optimization of the Synthesized Condition for Carfacrol Derivative. Next, my presentation has 4 contents. I will begin with a background and motivation. Then, I will jump into the experimental part, followed by spending some time on a result and discussion. Finally, I will discuss my conclusion. So, first of all, I begin by introducing the importance of carfacrol derivatization. Carfacrol is an aromatic monoterpenoid that can be found naturally in the various plants. Howe et al. 2021. It has various biological activities. Unfortunately, the use of carfacrol as a lead compound has limitation due to its hydrophobicity and volatility nature. Therefore, we need to do chemical modification of carfacrol to overcome the limitation. 5-isopropyl to methylphenyl to chloroacetate, hereinafter refers to as carfacrol ester during this presentation, is one of the examples of carfacrol derivative. This compound is a precursor for synthesizing carfacrol phenolic acid hybrid, Ashraf et al. 2017. This compound have activity as mushroom tyrosinase inhibitor. It can be used as a promising candidate for the development of safe cosmetic agent. Previously, the carfacrol ester can be synthesized from carfacrol and chloroacetyl chloride in the presence of triethylamine as a base and dichloromethane as a solvent for 5 hours. Ashraf et al. 2017. However, optimization of synthesized conditions was still needed to enhance the reaction yield as well as reducing time of reaction. Nargide et al. 2008 have already developed a greener, simpler, and a more rapid synthesized method for carboxyl ester with higher yield and purity. The method was assisted by microwave irradiation. Moreover, the study has already explored the utilization of microwave irradiation to optimize the reaction conditions in the formation of carboxyl ester. Now, we jump to the experimental part. Firstly, the reported method has already been conducted for comparison to demonstrate the advantage of proposed approach. This method was called a method A to investigate the effectiveness of microwave irradiation. We don't use triethylamine as the base. Virtually no effects of microwave were detected with the reaction using acetyl chloride in the presence of triethylamine. Miyazawa et al. 2009. Next, we continue with method B. This method has already been developed by Narkede et al. 2008. In this method, we use sodium hydroxide as a base and silica gel as a solid support under microwave irradiation. Evidently, silica gel can be used as a solid support in the imidation reaction under microwave condition. Moreover, chromatographic silica gel is also inexpensive and requires no special treatment or activation. Ojeda Porras et al. 2015. And the last method is a method C. We have already replaced a dichloromethane with tetrahydrofuran as a solvent, adopting Wang's method. Not only the yield was high, but also reaction time was reduced. Wang et al. 2018. This is the main reason to use this method. Now we are in the result and discussion part. As we can see in the figure 1, there are two main peaks in the GC chromatogram of the reaction product obtained by method A. The peak at a retention time of 8.025 minutes was expected to be carboxyl. Furthermore, the peak at the retention time of 15.194 minutes was assumed as carboxyl ester. To validate our prediction, we can see the MS spectrum of compound represented by peak at retention time of 8.025 minutes. Figure 2 shows a peak with M/Z value of 150.1, which corresponds to the molecular ion of carboxyl. Next, we also can see the MS spectrum of compound represented by the peak at a retention time of 15.194 minutes. Figure 3 also shows the peak with M/Z value of 226.1, which corresponds to the molecular ion of carboxyl ester. Unfortunately, when we attempt to synthesize carboxyl ester using microwave irradiation or method B, we only got one spot in the TLC plate. We have already proven that the compound is remaining of carboxyl based on proton NMR data. We didn't find not only chloroacetyl chloride in the reaction mixture, but also carboxyl ester as the expected product. Next, we continue with figure 7. Sorry. Now we continue with figure 6. The figure 6 show the GC chromatogram of reaction product obtained by method C. We also can see that there are two main peaks. The peak at the retention time of 8.729 are expected to be carfacrol, and the peak at the retention time of 15.497 minutes are expected to be carfacryl ester. The figure 7 also show a peak and M/Z value of 150.1, which correspond to the carfacrol. Figure 8 also show a peak with M/Z value of 226.1, which correspond to the molecular ion of carfacryl ester. The structure of carfacryl ester was also confirmed by proton NMR data. The singlet signal at 4.554 ppm of proton methylene is the typical signal of carfacryl ester. The typical singlet signal of carfacrol for OH proton also doesn't appear. The three signal at the chemical safe around 7 ppm are represent the aromatic proton in carfacryl ester. And the fourth signal at chemical safe around 1 until 3 ppm confirm the aliphatic proton in carfacryl ester. So this table shows the comparison of reaction for synthesizing carfacryl ester by using method A, method B, and method C. Based on the table, we can see that the good result is still obtained from method A by using dichloromethane as a solvent. Then these are our conclusion. The results show that microwave irradiation is not suitable to be applied in the synthesis of 5-isopropyl to methylphenyl to chloroacetate from carfacrol and chloroacetyl chloride. The applied high temperature and pressure was found to be unsuitable due to the volatility nature of chloroacetyl chloride. Alternatively, another attempt which is aimed to reduce the reaction time of the conventional methods is executed via utilizing different solvents. Unfortunately, tetrahydrofuran was also unsuitable as alternative solvent in conventional method due to its poor solubility power. And that concludes my presentation. I thank you all. I would really welcome any suggestion for furthering our research. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much, Bu Rini, for your nice presentations. Is there any questions from the audience for Ibu Rini? Okay, maybe one question from me. You already did 3 methods to optimize the synthesis for carfacrol derivatives. But the first method, I see that the reaction running for 5 hours, if I'm not mistaken. So is there any plan to reduce the time for the first method?
Okay, thank you for your question. I already tried to reduce using method B and method C, but it seems doesn't work well. So the first method is already a good. Maybe in the next I want to reduce the time by using another method. Thank you.
Okay, thank you for the answer. So, we come to the, thank you, Rudy, for your presentation and also for your answer. So we already finished, all the presenter already finished the presentation, but there is important moment lagi. So there is an important announcement from the committee. So the certificate will be able to generate through registration system on September 9. And then the information of the best presenter will be able in website on September 9. And please also upload your full paper to registration system for publication process. And publication payment only paid when the paper has been accepted. So that's the important announcement. So we come to the closing ceremony.
Assalamualaikum warahmatullahi wabarakatuh. Distinguished ladies and gentlemen, we have reached an end of the Earth Science and Mathematics International Conference 2022. Coming to the third part of my job to provide the closing remarks, since we have all be closing the door on lively and stimulating discussion. However, it is also a privilege and an honor to be entrusted with such an undertaking at a gathering of such eminent people from different parts of the world, academia, researchers, industry, and all contributed parties. As with previous week conference, this year event has been outstanding. Our mind have been assailed by a torrent of ideas, information, statistics, interpretation, and vision. And it will probably be a day or two before we can sift through them all to consolidate our own personal perspective. There is indeed plenty to reflect upon, and if this in any way enhances our individual and collective contribution to meet the post-pandemic challenges, then the conference can truly be adjudged a success. Finally, Excellencies, ladies and gentlemen, on behalf of you all, I would like to thank the Rector, Vice Rector, Dean, and Vice Dean of Mathematics and Natural Science Faculty, Universitas Negeri Jakarta. Thanks also addressed to our speakers, Scientific Committee, our co-host, University Technology Malaysia, all scientific organizations collaborate with us, sponsor, and Ministry of Education, Culture, Research, and Technology. Their presence has been invaluable and without any doubt has helped make the event a great success. We greatly appreciate the support we have received from the members of the media in covering our activities. It is very important that the views expressed here are disseminated to a wider readership and audience, and clearly this task has been in very capable hands. We are also grateful to all those who have been involved in the organization of the event, the organizing and technical committees. Excellencies, ladies and gentlemen, Science and Mathematics International Conference is a biannual event. We look forward to seeing you again in 2024 when we shall reconference and discuss another topical theme affecting research and education of science and mathematics. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Dulu bisa digunakan gitu. Terima kasih Bapak Ibu. Kemudian untuk makan siang sudah disiapkan juga di area-area gitu.
Once again, on behalf of the committee, we would like to thanks to all the presenters for the participation in SMIC 2022 and see you again in SMIC 2024.